And over to you, Domenica.
Thank you, Ben, and thanks everyone for being here. In my talk today, my presentation, I will talk about timing in a slightly different perspective from the previous speakers, in a more cognitive one. And specifically, the overarching question I'm going to address with the studies today is the following. So how does the human brain represent, encode, read out different duration? So what does it happen to neural circuits that are supposed to be engaged in temporal perception when I present participants, when I ask participants to discriminate between durations that belong to different ranges, let's say 2 and 300 milliseconds versus 1 or 3 seconds. And a theoretical possibility is that tuning mechanisms subserve duration representation. And these theoretical possibilities has been supported, has received empirical support from, for example, human psychophysics, that, for example, show that the repeated presentation of a stimulus of a given duration can bias the perception of a subsequent stimulus in a repulsive way. So let's say a 400 milliseconds duration is perceived longer if I adapted with a 200 millisecond stimulus. And this repulsive effect, this adaptation effect, has been interpreted as suggesting the existence in the brain of duration channels. Other empirical support comes from animal electrophysiology. So there are studies that show in sensory regions in cats and bats, or more recently in medial premotor cortex of monkeys, of cells that are actually responsive to stimuli that have a certain duration. So to address the question of, to test the hypothesis of duration tuning in the human brain, we ran 2 fMRI studies at 7 Tesla. And the hypothesis we tested was not just the tuning idea, but also we imagined that this neuronal unit that responds selectively to different durations are also orderly mapped in contiguous portion of the cortical surface as to form maps. Okay, and in the first of these experiments that were conducted mainly by Futini and Masamichi, we use a fairly standard temporal discrimination task. So we presented subject with 2 Gabor patches, and the subject task was to decide which one of the 2 Gabor patches was presented for longer time. And in different pairs of stimuli, S1 and S2, and in different trials, we tested different pairs, so different ranges. So ranging from 200 milliseconds up to a second. And we were, at brain level, we were actually interested in the brain response at the offset of the S1 duration, so, which is basically the offset of the purely encoding stage of the task, when basically the duration becomes finally available to the subject. And for this first experiment, we used the standard GLM whole brain approach. And what I'm showing here are group level results. So what you see are color-coded, are the vertices, so the voxel projected into the cortical surface, that were classified according to a winner-take-all procedure as maximally responsive to each of the different S1 duration. So the red vertices are describes response, maximum response to the shortest duration, to the shortest S1, and green to the longest S1. So as you can see, in 2 distinct brain regions, so in the supplementary motor area and in the intraparietal sulcus, we see tuning. But we also see topography, so because in both areas we see a nice spatial progression that goes from anterior to posterior in supplementary motor area and from lateral to medial for short to long in the IPS. So then, to better quantify the spatial progression of those duration-selective vertices, we computed several volumetric and surface indices, and one of these was the distance of each duration-selective vertex from one of the 2 borders of the map. And this is what I'm showing to you. So on the y-axis, you see the weighted relative distance from the posterior border. This is SMA, basically chronomap. And on the x-axis, you see the different duration-selective vertices color-coded as before. So basically what this plot, and this is, sorry, is the mean and the interquartile range and the fitted slope. So what this plot is saying is that the vertices, that the green vertices, so responsive to longest duration, are closer to the posterior border compared to the red vertices that are the farthest from the border. And this actually negative slope was present for the majority of the tested subjects, as you can see from the right-hand side. The results were actually a bit less nice in the intraparietal sulcus, where basically the expected positive slope was not observed in the whole individual subjects. Only 4 out of 11 show it. So basically, topographic maps were actually present in SMA, but not so much in individual subject in IPS. Then we decided also to look at duration tuning of these duration-selective vertices, especially to appreciate the response of those vertices to the non-preferred durations. And these are the results for the supplementary motor area. So you see this on the y-axis, the normalized signal change. On the x-axis are the different presented S1 durations, and the different lines are the different clusters, the different duration-selective clusters, color-coded as before. So everything is normalized to the preferred duration, and basically this plot shows that the bold response decreases, but moving far from the preferred durations. And this is basically happened in SMA, but also in the intraparietal sulcus. So basically in SMA, we see chronomap, so topography, and in both SMA and IPS, we see duration tuning. Right, we were happy with these results, but in order to prove the robustness of it, we decided to run a second experiment in which we use very similar stimuli, very similar tasks. So subjects still see 2 sequential Gabor patches, and they have to decide which one of the 2 was longer. But this time, we decided to use a broader range of S1 durations, so spanning from 200 milliseconds up to 3 seconds. And we also decide to introduce a second task. So we actually, in separate fMRI runs, so our subjects were either paying attention to the temporal dimension of the stimuli or to the spatial orientation of the Gabor patches. And this was done with the purpose of testing the task specificity of the chronomaps. And finally, we decided to present the pairs of stimuli, the pairs of durations, in a sequential manner. So we had ascending cycles where we started from the shortest pair and we ended with the longest pair, and in descending cycles we did the reverse. So as you might guess, this time we use a population receptive field model because the protocol was more suited to that. The PRF model we used was a unidimensional Gaussian with a certain mu, which was the preferred duration, and a certain spread, the sigma. And here are the results. So this is during the temporal task, so it's supplementary motor area. So we projected on the cortical surface the different mu, so the estimated mu parameters. The color coded is as before, so red is, so warmer color represents selectivity to shorter duration, and colder colors to longer. So we were able to replicate the original results, and this happened not only when subject was actually paying attention to time, but also when the features to attend was different, so was the orientation. Right, so when we look at the spatial displacement, so the relative distance from the posterior border in those maps, we noticed the following. So we saw that the negative slope was much better for durations below the second rather than above the second. And this was true for both hemispheres in the temporal task, but also for the orientation appropriate the temporal map in the orientation discrimination task, as if the spatial progression is better for durations shorter than a second than above the second. Right, at this point, I think it's worth emphasizing that those maps, differently, I would say, from sensory maps, were highly variable in the individual subject. And we wonder whether this high variability that we saw in the individual maps was linked to behavior of the subject. So we actually wanted to really link this goodness of the spatial progression as measured from the slope, so of the weighted relative distance, and two measures of subject behavior, so the accuracy and the precision of the temporal judgment. And we actually saw the following, so that in the first experiment, this is the result of the first experiment, we see this strong correlation between steepness of the slope and accuracy and precision of the spatial progression. So the better the spatial progression, the better the performance. And these results are very strong for first experiment, a bit less strong in the second experiment. We see the same trend, but this is probably due to the lack of statistical power, since we had less data point per conditions. Right, so then we actually we ask ourselves, but how is duration represented in these SMA chronomaps, in a relative or in an absolute fashion? And since we tested in different experiments 2 different ranges, we basically could compare the 2 maps. And this is basically the 2 maps overlaid on a same Dartel template. And as you can easily see from the image, by testing a broader range of duration, the maps stretch out anteriorly. And when we measure the absolute distance of each of the duration selective vertices from the posterior border, as you can see in this lower plot, where basically the circles represent the average distance of the vertices in the second experiment, and the square are the first experiment. So, and if you look at the vertices that are exactly the same tuning across the 2 experiments, which are the red, the orange, the yellow, and the green, you can, I think, appreciate that there was an anterior shift of those vertices. And actually, we also noticed that a really small percentage, so only 5% on average of those duration selective vertices kept the same tuning across the 2 experiments. So meaning really that the maps reshape according to the duration at hand. Okay, so, so the following question we try to address, but this is actually still an ongoing study that is led by Shrikant, is the question is, okay, are those chronomaps in SMA maps of perceived or of physical duration? So in order to address this question, we use basically an auditory task in which basically the same physical duration was perceived differently in 2 different temporal contexts. So we have the short and the long context that share the same 600 millisecond duration that was really perceived differently. And this was basically what we see so far, so we are still on the analysis stage. So in red are the vertices that corresponds to the 600 milliseconds when it's perceived shorter, compared to the blue vertices when this duration is perceived longer. So it looks like that although these results are still, again, a bit preliminary, I would say that it looked like that the map followed the same progression that we see before. So I would say that probably the maps reflect, or they are linked to perceived rather than physical duration. So, okay, so to conclude, I would go back to the original question. So how does the human brain represent different durations? And the answer is really, I don't know. I don't know yet. But of course, the data I presented to you today seem to suggest that duration representation is observed by duration tuning in SMA and IPS and by chronotopy in supplementary motor area. And what I also, the data show is that SMA chronomaps are linked to performance and perception and represent time in a relative fashion. So my interpretation of SMA chronomaps is that these are really a readout of time. So they represent at a high-level stage of temporal processing. It's where a duration is somehow stored in an abstract fashion and becomes available for motor action, for decisions. And so I should conclude by thank you for your attention, and for thank you all the people that made this work possible. Sorry, I just... let's go. Which are, okay, the people in my lab, especially Fotini, Protopapa, who are posted yesterday, Shrikant, all the people at CIBM Lausanne who helped me to collect the fMRI at 7 Tesla. Thank you.